Okay, so we're going to look at some samples using a microscope. But first of all, I thought, well, let's have a look at a traditional light microscope and see about the various components. So the bit that you look through at the top is the eyepiece. You look down through the main part of the microscope towards the turret, which holds the objective lenses, which are the ones that I'm twisting here. Now, this microscope comes with 4 different objective lenses. And usually on an objective lens, it says the magnification that they are. So this is times 5, times 12, times 40, and times 60. You can see the higher the magnification, the longer the lens piece is. And in fact, on this times 60 lens, it's actually, the objective lens is right at the base there. So you've got to be very careful you don't scratch it when you bring the lens down close to the sample. Now, the way you calculate the magnification of what you're looking at is actually the magnification of the objective lens times the magnification of the eyepiece. Now, this is a rather special eyepiece because it's actually got a zoom, so it actually goes from times 10, and if you twist it, it goes round to times 20. I'll show you that working in a minute. But that means that what you can do is you multiply times 10 at the top when it's on the 10 setting with the objective lens magnification at the bottom. And when it zooms up to times 20, the maximum magnification that this light microscope can at least achieve on paper is 60 times 20, so 1,200 times. That's really pushing the limit, actually. 1,000 times is pretty high for a light microscope. The sample is mounted on the stage, so you have these clips, and this is where you would put the slide that has the sample, and then you can actually illuminate the sample from below. There's a light here, a little bulb, which would turn on, and that shines the light through the bottom of the stage, and it's particularly useful, of course, for transparent or semi-transparent specimens. Otherwise, you need to rely on the light coming in from the sides in order to illuminate from the top to allow you to look at opaque specimens. And you use the coarse focus here to bring the objective lens down and focus on the sample, and then you can further tweak that with the fine focus there. Okay, so this is actually quite a nice microscope. It's actually one I had when I was a child, made by Prinz. So it's nice, metallic and robust. Now the problem is, of course, is you're not going to be able to look down there very easily. And in fact, this one doesn't come with attachments to allow you to plug it into a computer. It's too old for that. But what I'm actually going to use today is a USB microscope. Now these have come a long way, and this one's actually a few years old and is not expensive. It plugs into the USB port of a computer. It's basically a camera. It has some illumination around it. This one also has a zoom focus. Now, it doesn't have as great a magnification as the one I was showing you a minute ago, but it will go from times 10 to times 150. So it's perfectly adequate for the sort of things I'm going to show you today. And actually, you'll be quite amazed what you can see with just this modest magnification. Okay, so the first sample that we're going to look at, and this is something, of course, you can find at home. Just get yourself a bit of newspaper here. And what we're going to do is look at the ink on the newspaper and see how that looks. So here we are just on the plain white area of paper, which is fairly featureless. But as I move now towards the writing, you can see the black ink, the solid area of the writing. But what's most interesting is when you look at coloured areas. So I'm going to move across now to the dark blue. You can see that the dark blue is actually made up of dots. There you are. So it's not actually one solid dark blue colour, but it's actually made up of the 3 ink colours that you actually see in your inkjet printers maybe at home. You've got the cyan, the light blue, the magenta, which is the sort of pinky colour, and then yellow will be in there as well. And then it also looks like they're printing with a black, which of course is what happens in your printers at home as well. You've usually got a black cartridge. So in a very fine scale seen by the microscope here, you can see that the red, slightly off red colour, actually consists of dots, and those dots are yellow and magentas, and there's some black in there as well. If we go across to a lighter blue area, you can see that the cyan dominates with little bits of magenta and black to get the shade right, and then we can look maybe over here at some printed areas which are sort of orange and yellow. Okay, so the next 2 samples we're going to look at some rock. So if you go out into your garden, you may be able to find some interesting rocks. This one you can see it's actually got some shiny areas on it. It's probably some silica, some quartz on the surface of what otherwise looks like a fairly dull rock. So we're going to have a look at that. And the other thing you can obviously find out and about probably are some bird feathers. So again, let's have a look at what this looks like using the microscope. So when we look at the rock first, this was the sort of the browny edge that I showed you first. So you can see sort of sandstone in colour, and there's some features there. The microscope picks those up quite well. And then as we move round to that shiny surface, we can now focus in on these little areas of quartz or silica. You can see here the facets of those crystals, and these are now a lot clearer using the microscope. They're really quite amazing structures there. We're looking at the base of the feather. This is the bit where it would have attached to the bird. It's a sort of hollow shaft called the calamus, and that goes on up through the feather. This is the main shaft of the feather, you can see there. And coming off the central shaft are these little barbs, and they themselves have got structures on them called barbules and hooklets, which are what actually keep those barbs together and form the surface of the feather and help the bird fly. So you can actually zoom in with the microscope to its slightly higher magnification setting and actually see some of those features in a bit more detail. So there you are. You can see those little hooklets and the barbules on each of the barbs which hold those feather structures together. So I thought we'd have a look at Velcro. So I expect you've got some Velcro on your shoes or your clothes. As you probably remember, it kind of sticks together, but it's not glue. So the question is, well, why is it able to stick like that? You can tear it apart. What's the structure? So the microscope's a really good way of having a look at that structure and understanding what's happening. But one side, the fluffy side, is made of a tangled sort of mat of little fibres. So we're looking at that there. And then these are a bit like loops that are sticking out from the base of the Velcro material. But when we look at the other end, the sort of the rougher side, the firmer side, you can actually see maybe just about that there's actually a lot of little hooks, they're little plastic hooks, hook-shaped fibres that are sticking out of the surface. And it's these hooks that hook onto that messy mat on the other side that enable Velcro to stick. If you go to a higher magnification, you can sometimes see these hooks a little better. Now, it's difficult because they're obviously sticking out of the surface, and the microscope's only any good really at focusing on one plane. Basically, when you're ripping Velcro apart, you're pulling those hooks out of the tangled mess that's on the other side of the Velcro. I've actually got here some dead flies and dead ladybirds. So I didn't kill them, they were already dead when I collected them. But again, let's see if the microscope can show us some of the structures on these insects. Okay, so first of all, we're looking at the fly, and you can see it's actually quite hairy, and you can also see its eye to the top left of the picture. And its eye, of course, is a compound eye. It's got lots and lots of eyes. That's the fly's wing. And then the fly's body, again, lots of hairs moving down its legs as well. This is the underneath of the ladybird. You can see its leg, and the back of its shell, or the bottom of its shell. You go to a slightly lower magnification, maybe get a better feel for that. So you can see the underside of the ladybird here, with its 3 legs on each side, 6 legs in total, and then its head up to the top right. Here we are from the top now, its head, and then the shell underneath which are its wings that are hidden away in its spots. And you can see on the shell that there's some structure there, it's slightly dimpled. If we go to a high magnification, you can have a look at that in a bit more detail. As you can see, the surface almost looks like the surface of an orange. Next thing I want to do is get some pond water and have a look at a sample of that to see if we can see any pond life. Now, the best thing to do to look at that is to get some pond water into a little dish. Okay, so here we are now looking at the pond water sample. Now, it takes a bit of patience because you've got to go around, but you can often see things moving and swimming. So here are some odd things swimming around there. You can maybe just see towards the top right some sort of pond creature swimming, and there's some smaller ones that are also moving around under their own steam. So actually, the longer you look, you can see more and more creatures moving in the pond water sample. Just goes to show, all I did was scoop it off the surface, and there's lots and lots teeming with life.